Good evening, everyone. Now, again, absorption of plant physiology, absorption of water and mineral by the roots. Right. We have already discussed the vital function of the plants, and that we have discussed regarding the diffusion and osmosis. I've already given you the definition of osmosis. Full definition we have already done. Now we have to start with another topic starting from osmosis onward. That is, first thing we have osmotic pressure. Then we have DPD. Now, what is osmotic pressure? Then we talk about DPD. DPD is diffusion pressure deficit. DPD. You will get another definition of osmosis here on the basis of DPD, diffusion pressure deficit. Now, what is osmotic pressure? Osmotic pressure, it is the pressure developed in that solution. Now, when the osmosis is going on, when we have the two solution and through the semi-permeable membrane, osmosis is going on. So, what is osmotic pressure? It is a pressure exerted in that solution. Osmotic pressure, it is a pressure developed in that solution which can prevent osmosis in that solution. When the solution is separated from pure water by semi-permeable membrane. Again, I am repeating what is osmotic pressure. It is a pressure developed in that solution which can prevent osmosis in that solution. When the solution is separated from pure water by semi-permeable membrane. That is osmotic pressure. All right. Now, after the osmotic pressure, we have to go through DPD, diffusion pressure deficit. What is DPD? When you are adding a solute to a pure solvent, what will be the effect on diffusion pressure? Say the pure solvent or the pure water we are taking here. And in this pure water, we are adding what? Solute. Say a salt. When we are adding a solute to a pure solvent, then the diffusion pressure is lowered. Diffusion pressure is lowered. If you are adding the solute here, so here the diffusion pressure, dP is the diffusion pressure is lowered. The amount by which diffusion pressure is lowered by the addition of solute is known as diffusion pressure deficit. All right. So how you write the definition of diffusion pressure deficit? When the solute is added to a pure solvent, say water, it is a pure solvent. When the solute is added to a pure solvent, then the diffusion pressure is lowered. The diffusion pressure is lower. So amount by which diffusion pressure is lowered due to addition of solute is known as diffusion pressure deficit. All right. And always note one point, water always move from water. So this is another definition you get for the osmosis. Water always move from lower DPD to higher DPD. Lower DPD to higher DPD. Another definition of osmosis you can have. The water molecules always move from the lower DPD to higher DPD. All right. We have a relationship between the DPD. We have a relationship between DPD, osmotic pressure, turgor pressure, and the wall pressure. So this turgor pressure and the wall pressure we talk about after talking about osmosis in a plant cell. First, again, I'm telling what is the diffusion pressure deficit when you are adding a solute to a pure solvent. The diffusion pressure is lowered. All right. So by adding amount of solute, the diffusion pressure of a pure water or a pure solvent is lowered, is known as diffusion pressure deficit. Now, first we talk about osmosis in a plant cell. Very important topic. Board ask you the question from here. Reason-based question. First, understand osmosis in a plant cell. You have already studied in class 9 plant cell and animal cell. It comes under the eukaryotes. Eukaryotes are those organisms which have a definite, well-defined, well-organized nucleus. Right? Now, osmosis and plant cell. So we know in osmosis we have the cell wall. In osmosis in a plant cell, we have a cell wall as well as a cell membrane are there. And in the plant cell, we have the bigger, large vacuole consisting of, this is the vacuole. We have the cell sap, sugar or watery solution inside the vacuole that we know it. We have the nucleus also here. All right. Now, what will happen here? In the plant cell, we have the cell wall as well as the cell membrane. Cell wall is made up of cellulose. All right. So when you are putting this osmosis will be going on when you are putting this plant cell. Where you will put this plant cell? You will put this plant cell in pure water. Last time I've already told you, pure water is hypotonic solution. Write few lines here. When a plant cell, this plant cell is placed, when a plant cell is placed in pure water or hypotonic solution, then water enters into the plant cell. Water enters into the plant cell by end osmosis. When a plant cell is placed in pure water or hypotonic solution, water enters into the plant cell by end osmosis. When water enters into the plant cell, it develops a hydrostatic pressure. Here it is developing the hydrostatic pressure, water pressure. When water enters into the plant cell, it develops a hydrostatic pressure known as TP. That is turgor pressure. So in the board, they ask you what is turgor pressure. The exact definition of the turgor pressure is the pressure exerted by the content of the cell. Turgor pressure is the pressure exerted by the content of the cell on the cell wall. By the content of the cell on the cell wall is known as Tugger pressure. In name the following, they ask you pressure which is developed by the content of the cell on the cell wall. TP, Tugger pressure. It is a pressure exerted by the content of the cell on the cell wall. Now, what will happen when the Tugger pressure is applied by the content of the cell on the cell wall? Right there. When the Tugger pressure is applied on the cell wall, the cell wall being rigid exerts the pressure on the content of the cell. Now you will get another pressure on the content of the cell in opposite direction. On the content of the cell in opposite direction, you have the pressure. When the Tugger pressure is applied on the cell wall being exert the pressure on the content of the cell. This pressure is known as wall pressure. So the Tugger pressure is equal to wall pressure, which is equal in magnitude but opposite in direction. You can write the Tugger pressure and the wall pressure are in opposite direction but equal in magnitude. Therefore, the resultant pressure becomes zero. Therefore, the resultant pressure on the cell wall is zero. And the cell wall does not break and plant cell does not burst when they are placed in pure water or hypotonic solution. The board asks you the question. When the plant cell is placed in pure water or hypotonic solution, it does not burst. You simply write the answers there. When the plant cell is placed in pure water or hypotonic solution, the Tugger pressure is applied by the content of the cell on the cell wall, and the wall pressure is applied on the content of the cell. Both are equal in magnitude but opposite in direction. So the resultant pressure on the cell wall is zero. So the cell wall do not burst and the plant cell do not burst when they are placed in pure water or hypotonic solution. Again, another question then. What will happen instead of plant cell? If the animal cell is placed in pure water or hypotonic solution, all these are the board's question only. But the animal cell burst, write it there. But the animal cell burst when they are placed in pure water or hypotonic solution. Because animal cells have no cell wall. Animal cells does not have cell wall. So what is their outer boundary? Cell membrane. And you know cell membrane is a living membrane and the soft membrane. So, but the animal cell bursts when they are placed in pure water or hypotonic solution. Because animal cells does not have cell wall, they cannot counter the Tugger pressure. They cannot counter the Tugger pressure. So this Tugger pressure will go where? On the cell membrane. They cannot counter the Tugger pressure. The Tugger pressure is applied on the cell membrane and cell membrane breaks because it is very soft. Got it clear? So the Animal cell burst when they are placed in pure water or hypotonic solution. Note one important point here. When the plant cell is placed in pure water, they absorb water by the process of end osmosis. Here, the cell is said to be turgid cell, and the cell is said to be turgid cell. Turgid cell being rigid and hard, they ask you the state of the cell. And the turgid cell is stiff, and the state of the cell is called turgidity. And the state of the cell is called turgidity. Was it clear? Now you know why the plant cell, when placed in pure water, do not burst, and why the animal cell, when they are placed in pure water, burst. All right, because animal cell cannot counter this Tagga pressure. So this Tagga pressure is directly applied on the cell membrane, which is very soft and wet. So animal cell burst when they are placed in pure water. Was it clear? Now next heading we have the relationship between, you have already studied DPD, Tagga pressure, and the wall pressure. Let's talk about relationship between DPD, Tagga pressure, and the wall pressure. We have the relationship DPD, that is diffusion pressure deficit, that will be equal to osmotic pressure minus turgor pressure. Osmotic pressure minus turgor pressure. Here we have the osmotic pressure also, because these equal in magnitude but opposite in direction. So this is the DPD relation, osmotic pressure minus turgor pressure. Now note down the one of the numericals we have. There are two adjacent plant cell A and B. There are two adjacent plant cell A and B. These are the two adjacent A and B, in which the osmotic movement of water is going on. A has osmotic pressure of 20 atmosphere and turgor pressure of 16 atmosphere. B has osmotic pressure of 10 atmosphere and turgor pressure of 8 atmosphere. So what they have asked in the board? Find the direction of water. So what is the question now? Find the direction of water. You have to find the direction of water. Now, relation we have, dPd is equal to OP minus Tp. So for A, dPd will be, OP is how much? 20 minus 16, 480. B is having the dPd, 10 minus 8, 280. I have given you one point. That was the definition of osmosis. Water always moves from lower to higher dPd. So what will be the direction of water? The direction of water, lower dPd to higher dPd. So the direction of water is from B to A. This is the answer. Direction of water is from B to A after calculating the dP. All right, clear? Now, we have reason-based questions. On the basis of these osmosis, let's talk about which board has asked you, and these are the most important reason-based questions. The first one. Question number 1. Marine fishes burst. Marine fishes burst when they are placed under tap water. Marine fishes burst when they are placed under tap water. This is the question number 1. Question number 2. Potato tuber chips. We have potato tuber chips becomes stiff. Potato tuber chips becomes stiff, means hard, means rigid when they are placed in pure water. And the third, this is too much applied based question. When the fertilizer is added to the soil, now the farmers are adding the fertilizers, obviously. When fertilizers are added to soil, soil is adequately watered. These are the reason-based questions from ICSC. Marine fishes burst when they are placed under the tap water. Potato tuber chips become stiff when they are placed in pure water. And the fertilizers are added to the soil. Soil is adequately watered. Now, marine fishes burst and they're placed in tap water. You know tap water. What is tap water? Tap water is one hypotonic solution. Just now I've already told you. Animal cells burst. Marine fish, animals burst. Write it. Tap water is hypotonic solution. Therefore, water enters into the body of the fish. This is a keyword. Therefore, the water enters into the body of the fish by end osmosis. Without this, you won't get mass. So important thing. Tap water is hypotonic solution. And water enters into the body of the fish by end osmosis and develops turgor pressure. You have studied just now. Therefore, marine fishes burst when they are placed in tap water. Tap water is a hypotonic solution, and water enters into the body of fish by end osmosis. These two are the key words where you get mass. And develops turgor pressure. Got it? So marine fishes burst and they are placed in tap water. Now the second what we have, potato tuber chip become stiff when they're placed in pure water. So again, I have already told you, always remember, pure water is hypotonic solution. Pure water is hypotonic solution. Therefore, it enters into the potato tuber chip by end osmosis and it develops turgor pressure. So chips become potato tuber chips become turgid and stiff. Got it clear? Now the third fertilizers are added to the soil, the soil is adequately watered. Immediately, the farmers, when they are adding the fertilizer, they also have to add the water. You know why? When the fertilizer is added to the soil solution becomes, when the fertilizer is added to the soil solutions become hypertonic. Soil solution becomes hypertonic. When water is adequately added to the soil, then the soil solution becomes hypotonic. Means for when the fertilizer is added to the soil, at that time the soil solution becomes hypertonic. So soil is adequately watered, soil solution becomes hypotonic, and root hair will absorb water by end osmosis. If the farmer will not add the water here, this become hypertonic. How the root will absorb water? Instead of that, root hair water will go to the soil. So again, I'm repeating this. When the fertilizer is added to the soil solution becomes hypertonic solution. So it is adequately watered. Then the soil solution becomes hypotonic solution. So root will absorb water by end osmosis. Got it clear? All these are the reasons the board has asked you so many times these questions. Clear? Now, next topic what we have, experiment on osmosis. We have experiments on osmosis. Wait.